everyone, and welcome to episode 13, Remastered. This is the first episode in a series on genetics. Genes, alleles, transcription and translation, meiosis, all the fun stuff. Now this episode will introduce genetics by exploring the story of how humans developed their first formal study of genetics. It's kind of like how humanity had its first intro to genetics. To do this, I'm going to be exploring the experiments and the research of the Austrian monk Gregor Johann Mendel. Between the years 1856 and 1863, Mendel conducted experiments with pea plants. These experiments involved the reproduction of successive generations, where the traits that emerged in the phenotype were recorded and cross-referenced with their parents and grandparents. In this way, Mendel's work developed the rules that we use for understanding and explaining heredity. Collectively, these rules are called the laws of Mendelian inheritance, honoring their creator, Gregor Mendel. Now, he announced his work to the public in 1865, but it received little fanfare and attention for nearly 34 years. Unlike Darwin, Mendel's work was not enthusiastically appreciated by his contemporaries. It wasn't until much later, around the early 20th century, that experiments with more refined technology backed up almost all of Mendel's claims, giving his scientific literature validation as the brilliant work that it was. It was truly ahead of its time in many, many ways. Mendel was born to an Austrian family living in what is now known as the Czech Republic. His early life was on a farm, where he learned beekeeping and gardening. He studied at the University of Olomouc from 1840 to 1843, but suffered from both financial troubles and illnesses. In order to continue his studies without worrying about the expense, he joined the church and became a friar, receiving the name Gregor. He tried to become a substitute teacher at the Augustinian St. Thomas's Abbey, but failed. Having then studied physics at the University of Vienna, he came back to try to become a teacher, but failed again. In 1867, he turned his attention away from teaching and scientific experiments to become an abbot, which was a service that he performed until his death in 1884. It was at the University of Olomouc, early in his scientific career, that Mendel conducted his famous experiments on plant genetics. He studied the pea, using it as a model organism due to its rapid growth cycle, its abundance of seeds, and the minimal care that it required. He selected 7 traits about the pea plant that he wanted to study, to track and observe across multiple generations. The traits that he chose were the plant's height, the shape and coat tint of its seeds, the shape and color of its pods, and the color and location of its flowers. The species of peas that Mendel was working with had both male and female parts. They were hermaphroditic, and so they would normally self-fertilize. To put a stop to this, Mendel removed the male parts from the plants before they produced and released pollen in order to prevent this self-fertilization. Mendel then took the pollen in the male parts and brushed them deliberately onto specific female parts of other pea plants that he wanted to crossbreed with. This allowed him to control which plants mated with which, to select favorable or pure strains and create populations with specific traits. A trait is the phenotypic expression of an allele of a gene. There's a few important terms here that I should define. A gene is a small region of a strand of DNA which codes for a protein or a group of proteins integral to the expression of a particular trait. The trait is the actual real-world physical characteristic, as a part of the phenotype, or the exterior cosmetic and physiological appearance of the organism. Alleles are versions of a gene that code for the same trait, but just a different version of the trait. For example, everyone has a hair color and an eye color, and these are both traits. We all have genes for hair color and eye color, which code for these traits. The allele codes for the specific variation to that gene, to that trait. So the genes for the eye color trait have multiple alleles. One allele codes for blue eyes, another allele codes for brown eyes, another for green and hazel, etc. Similarly, the genes for the hair color trait have multiple alleles, one coding for blonde hair, one for black hair, another for red hair, one for brown hair, or some other intermediate color like strawberry blonde or ombre, and this can be applied to virtually every other trait where there's some degree of variation. Mendel's pea plants also had alleles for their traits. The first trait that he looked at was the shape of the pea seeds. Mendel took pea plants from two purebred populations, one that was homozygous for round seeds, and another that was homozygous for wrinkled seeds. Now I've just introduced a couple more terms that I should explain and define. So purebred, or a pure genetic line, means that the individual will produce another individual almost identical to it if it's crossed with another individual from that same purebred population. Homozygous means that the individual has the same allele for each copy of the gene. Remember that diploid species have cells with two copies of the genome. They thus have two copies of each gene, and when each gene has the same allele, the individual is homozygous for that trait. In contrast, if each allele is different, then the individual is heterozygous for that trait. So Mendel crossed a purebred round seed pea plant with a purebred wrinkled seed pea plant. The offspring of this cross, to much surprise, all had round seeds. This discovery was really important because it meant that the traits did not blend together equally into some kind of intermediate, as was predicted by the prevailing theories of the day. The discovery also posed a question. Where did the wrinkles go? To find this out, Mendel had the offspring generation, or the F1 generation, he had them self-pollinate themselves. Mendel reasoned that some genetic determinant existed for both round and wrinkled seeds, and that both determinants existed in the F1 generation, even though only one of them, the trait for round pea seeds, was actually being expressed. Interestingly, the F2 generation, or the offspring of the self-pollinated F1 generation, the F2 plants had both round and wrinkled seeds in a ratio of 3:1. This means that out of every 4 offspring, one of them had wrinkled seeds, while the rest had round seeds. The importance of this ratio will become apparent when I talk about the mathematics of heredity. Based on the results of both the parent crossing and the F1 self-pollinating, Mendel reasoned that some traits were recessive, while other traits were dominant. I feel like it's very important to clarify that when we describe genes as recessive or dominant, we're not talking about their fitness benefits. We're not saying that a dominant gene makes the organism more dominant. We're not saying that a recessive gene makes the organism more recessive, or more submissive, or less fit, or whatever it happens to be. This is specifically talking about gene expression. The dominant gene is simply the one that gets expressed, while the recessive gene is the one that doesn't get expressed when it's in the presence of a dominant allele that codes for the same gene. Now, in the case of Mendel's peas, the wrinkled seeds was a recessive trait, while the round shape of the seeds was dominant. As for all of the other genes and traits in the pea plant that Mendel studied, they all had their own alleles as well. Seed color could be yellow or green. Yellow was dominant, while green was recessive. The seed pod shape could be inflated or constricted, with the inflated shape being dominant and the constricted shape being recessive. The seed pods could also be different colors, green or yellow, where green was dominant, while a yellow pod color was recessive. As far as flowers are concerned, purple flowers are dominant, while white flowers are recessive. The allele that positions the flowers axially was dominant, while the allele that positioned the flowers at the ends of the branches was recessive. Lastly, it seemed that taller height was the dominant allele, whereas dwarfish height in the pea plant was due to recessive alleles. Although height is kind of an interesting one. Height is what they call a quantitative or a continuous trait, because it's not that you have one trait with a particular allele that says, I'm the tall allele, you're now tall. It's more like you have a large number of these genes that promote height, and if you have more of them, you'll tend to be taller, and if you have less of them, you tend to be shorter. But of course, this is some really nuanced genetic biochemistry and chemical structure that Mendel had no idea about during his time. They had no way to understand any of the atomic structure of these things with the technology of the day. How did Mendel explain the disappearance of the wrinkled seeds in the F1 generation, and then their reappearance in a quarter of the F2 generation? His reasoning was ahead of its time. Mendel figured that each pair of genes must be separated into separate gametes. In this way, each gamete possesses one allele for each gene in the organism's genotype. As humans are diploid, we have 2 copies of each of our genes. When our chromosomes are divided during meiosis, 1 copy of the genome goes to each daughter cell, and so each daughter cell only possesses 1 particular allele of a particular gene, because they don't have that duplicated copy that might have a different allele. Each gamete is haploid, because they only possess a single copy of the genotype. This is a concept called the principle of segregation, and I'll talk more about it when we get to the episode on meiosis. So let's consider the first parent pea plants that Mendel used. These were purebred strains that varied in one characteristic: seed shape. Because round seeds are dominant, we can represent this allele for round seeds with a capital R, and call it big R. As wrinkled seeds are recessive, we can represent the wrinkled seed shape with the allele of a lowercase r, and just call it little r. So the parent that has purebred round seeds can be described as having the genotype big R, big R. They're homozygous for the dominant allele. They do not possess the allele for wrinkled seeds. Now the other parent is the opposite. They're purebred for wrinkled seeds, and so their genotype can be described as little r, little r. Now both big R and little r are homozygous genotypes. They possess the same allele on each copy of the genome. The F1 generation, however, possessed both a big R and a little r, which they each got from their respective parents. Every single F1 plant had both alleles for round and wrinkled seeds, big R and little r. But because big R is dominant, only big R got expressed, and thus the seeds of the F1 generation were only round. Mendel argued that each allele, was separated into individual gamete cells, alongside 1 copy of every other gene in the genome. So when the heterozygous F1 generation self-pollinated, each egg could possess a little r or a big R allele for the seed shape gene. Similarly, each sperm from the males could possess either a little r or a big R allele for the seed shape gene, and they could bind with any particular egg to make one of several combinations. The offspring of the heterozygous self-pollination of the F1 generation would exhibit 3 combinations of alleles. They would either have big R, or little r, and they'd be homozygous in these cases, or they'd be heterozygous with a big R, little r offspring. Now the big R offspring will have round seeds, as they only have the dominant alleles for the round seeds. And meanwhile, the little r offspring will have wrinkled seeds, because they only have the alleles for wrinkled seeds. But the heterozygous big R offspring are much like their F1 parents. They have both types of alleles, but only the dominant one gets expressed, so these have round seeds. You can visualize this genetic crossing with a Punnett Square. Imagine a square, and inside of this square are 2 crossing lines, splitting the square into 4 smaller boxes. On top of the square will represent one parent, so let's say the mother. So let's say that this is little r up here. And then on the left side of the square we'll have the father and his genome, and so we'll say that that's big R. Within the Punnett Square, you start to cross-reference all of these and see what combinations each allele would make if it was paired with one of the other alleles from the other parent. So let's look at a Punnett Square filled in with the information from Mendel's F1 generation. These F1 generation pea plants are all heterozygous for the seed shape alleles. They all have a seed shape genome that goes big R, little r. because they've got both traits, but the dominant one is getting expressed, and that's why the F1 generation all has the round seeds. So if you put big R little r on top of the Punnett square, and big R little r on the left side of the Punnett square, and you cross-reference them all, and fill in the Punnett square, you'll see that the top left square is big R, big R. So before I get into the specifics, you should notice that each square represents the genome of approximately 25% of the offspring. So say we had two F1 generation pea plants. We bred them together and we made 100 offspring plants. About 25% of these, so about 25 of them, are going to have the big R genotype with respect to their seed shape. And this is because if you look at the Punnett square, you'll see that that top left square has two big R alleles. Now if you look at the bottom right square, which is on the opposite corner from our big R genotype group, you'll see another 25% of the offspring. And these will have the little r genotype, so they'll have wrinkled seeds. Now if you take a look at both the upper right and the lower left boxes of the Punnett square, each of these boxes will have a big R heterozygous genotype, and each box represents 25% of the possible offspring. So these offspring will be heterozygous, but because big R is dominant, they'll express round seeds in their phenotype. And because these heterozygous offspring are represented by two boxes, they'll combined make up 50% of the offspring. So Mendel's F1 self-pollinating plants gave birth to 7,324 offspring. 5,474 of these had round seeds, while 1,850 of them had wrinkled seeds. Now Mendel did not have access to technology that would have enabled him to examine the genes on the chromosomes themselves, but his experiment demonstrated the 3-to-1 ratio, where about 25% of the offspring expresses the recessive trait and 75% of the offspring expresses the dominant trait, either because they're heterozygous and they have the dominant and recessive allele, or because they're homozygous and they just have the dominant alleles. This is a pretty simple, rudimentary demonstration of how alleles are split up from parents to offspring. These experiments were a monohybrid cross, which is a cross that's performed between two parents who differ in only a single trait. So besides the alleles for their seed shape, these particular lineages of pea plants that Mendel was using for this particular experiment, they had all the other traits were the same, so this was the only trait variance that he was studying. Now naturally, this raises the question, what would happen if you made a dihybrid cross, or a cross between parents who had two differing traits instead of just one? Consider the trait for seed color that Mendel studied. The dominant allele was for yellow-colored seeds, which is represented by a big Y. The recessive allele was for green-colored seeds, and that's represented by a little y. Now consider two hypothetical parents, one that has round yellow seeds, and the other that has wrinkled green seeds. So this is a dihybrid cross, and both traits are different in both parents. This will produce an F1 generation of offspring that are all heterozygous for both traits. Each allele exists in each individual offspring, but only the dominant alleles are being expressed. So in the case of our hypothetical dihybrid cross, the F1 offspring would all have round yellow seeds, as this is the representation of the dominant alleles and their heterozygous genotype. Now the really interesting stuff will start to happen when the F1 generation is self-pollinated. The F1 offspring, called the F2 generation, will have alleles that can do two possible things. During meiosis, they can be assorted independently into gametes, or they can be sorted depending on where one particular allele goes. The former possibility is called independent assortment, while the latter is called dependent assortment. It's pretty easy to keep track of these. Now, if the alleles were to be organized according to dependent assortment, that would mean that the alleles for seed shape are somehow associated with the alleles for seed color, or vice versa, such that when one of these alleles gets moved into a gamete during the chemical process of meiosis, the other allele that's associated with it is brought along, and it also goes to that same daughter cell. As a result of this dependent assortment, the offspring would all share the same group of traits, because these traits are shuttled as a group from parent to offspring. The hypothetical dihybrid cross would produce results that are very similar to our monohybrid cross. You would get 25% with all dominant alleles, 25% with all recessive alleles, and another 50% with heterozygous alleles for each trait. However, when Mendel made his dihybrid cross and self-pollinated the offspring to create an F2 generation, this is not what he found. What Mendel found was instead far more supportive of the concept of independent assortment. This is to say that during the formation of each genotype that would be given to each daughter gamete during meiosis, the alleles were sorted independently of one another. It does not matter, for example, where the R alleles go. The Y alleles will be sorted independently of the big R. They have an equal chance of going to the same daughter cell or the other daughter cell. And this is true for every allele. If we were to map this out with a Punnett Square, it would need to have 16 boxes, so 4 to a side. The tops of each column and the left sides of each row would be represented by one possible copy of the genotype that the gametes could receive from each parent. So the parents being from an F1 generation would be heterozygous for both traits, which means when they produce gametes through meiosis, 25% of these haploid gametes are going to have the big R, big Y genotype, and so they're going to pass down only dominant alleles. 25% of the produced gametes are going to have the big R, little y genotype, so these are going to pass down the alleles for round seed shape, but a recessive allele for color. Another 25% of the gametes are going to have the little r, big Y genotype, and then another 25% are just going to have the little r, little y genotype. These would only pass down the recessive traits, because they're haploid, they're only carrying recessive traits, so if they fertilize with a gamete that's heterozygous for some of these traits, then the dominant alleles that heterozygous one is carrying are going to be expressed, because the only alleles this one is carrying are recessive. So with each genotype written along the top and left sides of the Punnett Square, we can begin combining gametes to see the possible genotypes that could exist in the offspring. Mendel performed this exact experiment, getting 556 total offspring in his F2 generation. 315 of these offspring, or about 9/16 of the total, expressed the dominant alleles with round yellow seeds. 108 of these offspring, or about 3/16, had round seeds that were green in color. 101 of them, or close to another 3/16 of the offspring, had yellow color but wrinkled shape. These two groups of offspring are heterozygous for one gene, where the dominant allele is expressed, and they are homozygous recessive for the other gene, where the recessive allele is expressed. The final 1/16 of the offspring, which is about 32 individuals in this total population of 556, about 32 of them were homozygous recessive for both traits, expressing a phenotype with green color and a wrinkled shape. This 9:3:3:1 ratio in the dihybrid cross is like a mathematical square of the 3:1 ratio in the monohybrid cross. It also demonstrates that the principle of segregation in monohybrid crosses can translate in dihybrid crosses into the principles of independent assortment. Mendel tested the principle of independent assortment in another way, by using what's known as a test cross. A test cross involves a parent homozygous recessive for all alleles, and then a parent with an unknown genotype. The offspring of such a test cross can reveal information about the parent with an unknown genotype. So the parent with the homozygous recessive genotype, recessive for all alleles, will always pass on recessive alleles. That's all they have. And so if there's any dominant alleles that are expressed in the offspring, then you can deduce that these came from the parent with the unknown genotype. More specifically, if the parent with an unknown genotype is heterozygous for this or that allele, then it'll produce gametes with both dominant and recessive alleles. The offspring has a 50% chance of inheriting either the dominant or recessive allele, depending on which allele is in the gamete that fertilizes the egg. If the parent is homozygous for these dominant traits, then it will just produce gametes that have the same dominant alleles for each gene. Every gamete will have that dominant allele. The parent with the unknown genome can also have homozygous recessive alleles, in which case this will just be expressed as the recessive trait in the offspring. If a parent is homozygous for a particular gene, that is to say that they have the same allele on both copies of their chromosome, then with respect to that gene, with those alleles, they can only make gametes that have that allele. And so whether it's dominant or recessive, their offspring is guaranteed to have it, because there's no alternative allele for that gene that's available from that parent. Mendel's homozygous recessive parent had the genotype r, y. It was homozygous recessive for both genes, and its phenotype was a green, wrinkled seed. The other parent plant that Mendel used had an unknown genotype. However, he could observe the pea plant's phenotype, which expressed round yellow seeds, and he could kind of deduce what was about to happen. From these observations, Mendel reasoned that the unknown genotype must possess at least one big R and one big Y, out of 4 possible alleles. He's reasoning that there has to be at least one dominant allele in the genes here, because the phenotype is expressing dominant traits. So there's 2 possible outcomes to this test cross that Mendel created calculations for. The first possible outcome was if the unknown genotype was homozygous dominant for both genes, in which case every offspring would receive one dominant allele from that parent, and one recessive allele from the parent that is known to have homozygous recessive alleles. Every offspring would therefore have the same genotype, which has heterozygous alleles for every gene. This is because one parent could only give the offspring recessive alleles, and the other parent could only give the offspring dominant alleles, so every offspring has one of each. They're all heterozygous. Now, the second possible outcome is if the parent's unknown genotype is itself heterozygous for both of the genes. In this case, the offspring population could be divided into quarters, one having round yellow seeds, another quarter having round green seeds, the third quarter having wrinkled yellow seeds, and the last quarter having wrinkled green seeds. This represents every possible combination of alleles, equally represented in the offspring population. This demonstrates the validity of the principle of independent assortment. Now recall that at the beginning of the episode, I mentioned how Mendel's work wasn't really appreciated for its worth until the turn of the 20th century. The first work that validated Johann Mendel came in the form of a hypothesis published in 1902 about what else but meiosis. 2 scientists working independently of one another came up with the hypothesis that meiosis could explain both principles that I discussed a few moments ago: the principle of segregation that was demonstrated in the monohybrid cross, and the principle of independent assortment that was demonstrated in the dihybrid cross. I'll spend the entirety of the next episode talking about meiosis in great detail, but for now, all you need to know about meiosis is that it's a process by which a diploid progenitor cell makes copies of its pairs of chromosomes and then divides into 4 haploid gametes, with each of the 4 gametes getting one set of chromosomes from the 2 pairs that have been produced by the parent progenitor cell. These 2 scientists who worked independently of each other were named Theodore Boveri and Walter Sutton. They identified Mendel's so-called genetic determinants as specific loci on the chromosome. You see, the chromosome is a giant, coiling, piled-up mass of a DNA molecule, which is like a massive string that's been supercoiled. Portions of this string that code for proteins are called genes. The gene exists as a discrete stretch of the DNA, somewhere along the greater strand, so somewhere in the chromosome. This point on the DNA strand, or this little sequence where the gene is this point on the DNA strand, or this little length of sequences, wherever it happens to be on the DNA strand, is called a locus, or a loci to be plural. In Mendel's experiments, it was the loci for the various traits, like round shape or green color, that were scattered between gametes during independent assortment. During independent assortment, It's assumed that genes are on different chromosomes. If genes were on the same chromosome, then they would demonstrate some degree of linkage. Sister chromatids are pulled apart during a middle stage in meiosis, each one of them getting pulled to the ends of the parent cell to be integrated into one of the emerging daughter gamete cells. The genes that are on different chromosomes are pulled independently of each other, because one chromosome can always go one way and a separate, distinct chromosome can always go another, so they're independent. But when you're talking about genes and loci that are on the same chromosome, that's where things get a little bit messy. There was a man named Thomas Hunt Morgan, who was an American scientist, an evolutionary biologist and geneticist, who did his work in the early 20th century. Thomas Morgan conducted research on, among other things, meiosis in a model organism called Drosophila melanogaster, aka the common fruit fly. One of Morgan's experiments with the Drosophila looked at the linkage of genes on the same chromosomes, and how they moved during meiosis. His experiment looked at two genes on the creature's X chromosome. One of these genes, called W, was for an eye color trait, and another gene, called Y, was for a body color trait. The dominant alleles, big W and big Y, code for red eyes and a gray body. The recessive alleles, little w and little y, code for white eyes and a yellow body. In the experiment, he used individual flies with two different types of genotypes on their X chromosome. The females had two X chromosomes, one of them with little w, big Y alleles, and another with the big W, little y alleles. These females were mated with males who possessed a Y chromosome, as all males do, and an X chromosome with the little w, big Y allele. I should point out that the Y chromosome is just the male sex chromosome. It doesn't have anything to do with the big Y, little y alleles in this example. After these flies mated, they produced an F1 generation. Morgan looked at the genotype of the F1 generation to see if the alleles combinations on any X chromosomes had changed. Morgan predicted that if each gene was tightly linked together, he would expect the offspring to only have big W little y or little w big Y allele combinations. He wouldn't have expected them to change. However, if there was a degree of recombination within the chromosomes, it's possible that the alleles would get shuffled around. The offspring would then have some X chromosomes that had little w little y or big W big Y allele combinations, something that's unique from the parents. What Morgan found was that a large majority of the F1 offspring had the same allele combinations as their parents. But to his surprise, he also found that there was a minority of the offspring that had an X chromosome that had a little w little y or a big W big Y allele combination. He called this minority of the offspring recombinant, as they represented allele combinations that were unique and different from those in their parents. The natural conclusion to this experiment was that genes on the same chromosome don't always stay together when they get sorted into gametes. When I say they don't always stay together, what does that mean? How often do the genes not stay together? And how does that process work? This process by which genes can be shuffled around is called crossing over. During a stage in meiosis, the homologous chromosomes will swap huge chunks of their DNA with each other. As these homologous chromosomes carry copies of many genes, they only vary in the specific alleles at each gene locus. So when two homologous chromosomes perform crossing over and they swap segments of their DNA, the chromosome isn't being radically changed with respect to genes. It's keeping the same genes on the chromosome, but it's just swapping around the alleles for those that are on the other chromosome. Now, genes that are located on the sex chromosomes, the X and Y chromosomes, are said to be sex-linked. Genes that exist on the Y chromosome will only be expressed in males, as only males have the Y chromosome. The genes that exist on the X chromosome can exist in both males and females, as both males and females have at least one X chromosome. Males have one X and one Y chromosome, while females have two X chromosomes. Now, recessive alleles on the X chromosome will be expressed more in males than in females, as males don't have another X chromosome with a possible dominant allele to override the recessive allele. So think of it this way. Every cell in your body is diploid, so it has two pairs of each one of these 23 strands of DNA. So this means that ultimately, every cell in your body possesses 46 DNA molecules. 23 of these chromosomes, or one full copy of the genome, come from your father, and another 23 chromosomes, or the second copy of the genome, come from your mother. Every cell in your body has these 23 pairs of chromosomes times 2, one from each parent. The chromosome pairs themselves are numbered, as they contain genes for the same traits from person to person. For example, every human has a gene that codes for the protein titin on chromosome 2. Titin is a small, spongy molecule in the muscle tissue that gives it a degree of elasticity. Now, another example is a gene that codes for a chloride channel protein called CFTR, and that sits on chromosome 7. Now, if you were to have a mutation or an error on chromosome 4 involving the uncontrolled replication of the CAG nucleotide sequences, that'll cause Huntington's disease. Now, I could go on with more and more examples, but the point that I'm trying to make here is that all human genotypes are roughly analogous. The same genes for the same traits are in the same places on the same chromosomes in the nucleus of the cell. The reason there's so much structural consistency between the arrangement and placement of genes in the chromosomes of the human genome is because no matter where we are, all over the world, we're all the same species. The human species, Homo sapiens. During a stage in meiosis, these homologous chromosomes are lined up together, such that they can engage in crossing over. They can swap out segments of their DNA with each other. In this way, alleles that come from your father may be transplanted onto the chromosome copy that came from your mother. Your mother's alleles for the same genes would then necessarily have been transplanted from her chromosome copy onto the copy that came from your father. This is where the locus position on the chromosome becomes really important. The closer the two loci are to one another, the more likely it is that they'll stick together and be sorted together. In this way, they're linked. There's a linkage between the two loci. But loci that are far apart from one another on the chromosome don't necessarily share this proximal linkage. If the distance between them is great enough, it's quite likely that any given recombination event will just so happen to shuffle them around, or make them recombinant. We've created genetic maps of the chromosome, where we've literally mapped out the loci, where on the chromosome specific genes are located. In fact, we can use the distance between two loci on a chromosome, measured in nanometers, to determine the percent frequency of crossing over events that'll happen between them. Generally, the percent frequency of a particular loci crossing over from one homologous chromosome to the other increases the further from the center of the chromosome that it is. So the closer a loci is to the tip of an arm on the chromosome, the more likely it is to be swapped over in a recombination event. And this makes sense, as a recombination event will swap all DNA past the point of incision, past the particular point where the DNA was broken to initiate the recombination. It should be explained that not all genes operate on such a discrete level such as this in my examples. Not all traits in your body are coded for by a single gene or by a proximal group of genes. These traits that are coded for by a single gene or by a proximal group of genes, we call these discrete traits, because the trait is quite literally discrete. If the trait is coded for by a single gene, and you have one allele for that, but you switch the allele, or you just look at a different allele, and the trait is different, that's a discrete trait. It's different depending on the allele. But some traits, on the other hand, are coded for by a larger number of genes, and these large number of genes might be scattered across multiple chromosomes. These are called quantitative traits. Now, human height, for example, is a famous quantitative trait that's determined by many different genes on many different chromosomes, as well, of course, as environmental influences like diet and stress. These quantitative traits are passed on through a form of polygenic inheritance, where each gene will contribute a little bit towards the qualia, like height. So the more genes you have that contribute towards height, the taller you'll be, and vice versa. Now we're almost done, but not quite yet. There's still a little more detail that I want to squeeze out of the basics of this lesson. So I've talked about genes being dominant and recessive, where heterozygotes express one allele over the other. But not all alleles are so easily labeled. Some alleles are codominant. This means that two different alleles for the same trait are simultaneously expressed. This is the case for the AB blood type, where the individual simultaneously expresses the alleles for the A and B sugars that coat the surface of our erythrocytes, of our red blood cells. Additionally, some genes show a kind of incomplete dominance. In this circumstance of incomplete dominance, two alleles are expressed alternatively. Some cells will express one allele, while other cells will express the other allele. It's an incomplete dominance. This will create a mosaic pattern among the cells, which appears to us and our giant macroscopic eyeballs as a mixed or a blended hue. For example, consider the genes that code for pigments in human skin, or for the genes that code for the pigments in the petals of a flower. In humans, if a parent couple is biracial and possesses significantly different skin tones, then their offspring will receive genes for these different skin tones from each parent. Some skin cells will express the high melanin concentration of the darker parent, while other skin cells will express the low melanin concentration of the paler parent. When we see this cellular mosaic from our macroscopic, human-sized perspective, the skin tone appears to be a roughly equal mixture of mother and father. This same principle applies to flower petals. Some species of flower, for example, can have white or pink petals, and this color is due to the presence, or lack thereof, of a particular pigment in the cells. This pigment is not expressed in the white flowers, but it is expressed in the pink flowers. So if you have a plant with pink flowers, and it reproduces with a plant with white flowers, the offspring will have flowers that are a pale pink or a very light pink, because it's a mix of the two parent hues. Some of the cells in the offspring's petals will express the pigment, while other cells in the petals will not express any pigment at all. And again, on a cellular level, this looks like a mosaic or a patchwork quilt. And on a macroscopic level, the mosaic blends together to produce a shade of light pink. Furthermore, the genotype does not strictly determine the phenotype. The genotype definitely plays a role, but so does the environment. Consider, for example, a gene that breaks down a particular biomolecule. If you eat foods that are rich in this biomolecule, then it'll enter your system. And if you have the gene for this protein that breaks down the biomolecule, and that gene works, you'll produce the proteins that can break down and digest the biomolecule. It's pretty straightforward, it makes sense. But if your gene is faulty, or it's mutated and it doesn't work, then you won't be able to make that protein. And because you won't be able to make that protein that breaks down that biomolecule, you won't be able to break down the biomolecule. So if you eat foods that has that biomolecule, instead of being broken down, it will accumulate in your tissues and potentially cause problems like gastrointestinal distress. Or, in the case of the genetic disease PKU, it'll cause neurodegeneration. By adjusting to your environment, in this case by changing your diet to eat foods with a low level of the offending biomolecule, you can theoretically avoid these problems. Okay, so that is about it for this podcast. This was a long one. It went on way longer than I was expecting. There's so much information here that I could present, but I'm already going on longer than most episodes. There's so much detail, and there's so much to explore, and there's so much to explain. There's no hope I have of getting to it all. And I tried to put a lot of it in here, but, still kind of ran over time. Anyway, I hope you enjoyed this podcast, and I hope you learned something cool. In the next episode, I'll be discussing the process of meiosis in great detail. So if you're interested, then be sure to stay tuned for that. And as always, thanks for listening.